What if the very first living organism in the universe wasn't born on Earth at all? Picture a time before planets, before stars, even before light itself had fully taken hold. A universe so young and chaotic that the rules of life as we know them hadn't yet been written. This is where our story begins, not with a bang, but with a whisper, a single spark of life in the cosmic dark. How did it happen? Where did it come from? And what does its existence mean for us today? These questions lie at the heart of one of science's greatest mysteries, the origins of life itself. We call this puzzle abiogenesis, the moment when non-living matter somehow crossed the threshold into life. It's a mystery that has haunted scientists, philosophers, and dreamers for centuries. Think about it, every living thing on Earth, from the tallest tree to the tiniest microbe, traces its lineage back to that first unknown organism. But here's the twist, we still don't know what it looked like, how it survived, or even where it called home. Was it a simple chemical reaction in a bubbling pool of primordial soup? A cosmic traveler hitching a ride on an asteroid, or something so strange we haven't even thought to imagine it yet? That's where the theories come in. Over the years, scientists have proposed countless ideas, each one more fascinating than the last. Some sound like they're ripped straight from science fiction, while others are so elegant they feel inevitable. And today, we're diving into 30 of the most compelling, from the wildly speculative to the startlingly plausible. You'll hear about deep-sea vents that act as natural laboratories, ancient RNA molecules that might have been the universe's first code, and even the possibility that life didn't start here at all. This isn't just a history lesson, it's a detective story spanning billions of years, with clues hidden in the chemistry of comets, the geology of distant planets, and the very building blocks of our cells. So settle in, because by the time we're done, you'll never look at life, the universe, or your own existence the same way again. Let's start with one of the most daring theories of all, Panspermia, the idea that life didn't actually begin on Earth but instead hitched a ride here from somewhere else in the cosmos. Imagine a tiny microbe frozen in the icy heart of a comet, surviving the vacuum of space for millions of years before finally crashing into our young planet. That microbe, if the theory holds, could have been the seed of everything we know as life today. The concept isn't as far-fetched as it sounds. We've already discovered extremophiles, microorganisms that thrive in Earth's harshest environments, from boiling hydrothermal vents to the crushing pressures of the deep ocean. Some can even survive the radiation and vacuum of space, as experiments on the International Space Station have shown. If these tiny life forms can endure such extremes, why couldn't they make an interstellar journey? Famous astronomer Fred Hoyle was one of Panspermia's biggest advocates. He argued that space isn't empty, it's filled with organic molecules and possibly even dormant microbes riding on comets and cosmic dust. Some scientists point to meteorites like the Murchison meteorite, which crashed into Australia in 1969, carrying amino acids, the building blocks of proteins. If the ingredients for life can survive the trip, why not life itself? Panspermia also raises a thrilling possibility that life might be common throughout the universe. If microbes can travel between planets or even star systems, then life could be spread like cosmic dandelion seeds, taking root wherever the conditions are right. Earth might not be life's birthplace, just one of its many homes. Of course, this theory doesn't solve the mystery of how life began. It just moves the starting line somewhere else. But the thought that we might all be descendants of interstellar travelers, that's a story worth telling. Now let's plunge into the depths of Earth's ancient oceans, where another groundbreaking theory suggests life might have sparked into existence. Picture a world 4 billion years ago, our planet still young and volatile, its surface bombarded by asteroids and scorched by ultraviolet radiation. The safest haven, not on land but deep below the waves, where towering hydrothermal vents spewed superheated, mineral-rich fluids into the inky blackness. These underwater chimneys, known as black smokers, could have been the perfect crucibles for life's earliest chemistry. Here's why scientists find this idea so compelling. These vents aren't just geological oddities, they're natural laboratories. Their porous rock formations create countless microscopic chambers, each acting as a tiny reactor where simple molecules could concentrate, interact, and eventually assemble into something far more complex. The vents also provide a steady energy source, not from sunlight, which couldn't penetrate those depths, but from chemical reactions. This process, called chemosynthesis, powers entire ecosystems around modern-day vents, where bacteria convert sulfur and methane into energy. Could the same mechanism have jump-started life itself? Experiments back this up. Scientists have recreated vent conditions in labs, mixing water, minerals, and gases like hydrogen and carbon dioxide under extreme heat and pressure. Astonishingly, these simulations produce amino acids and even primitive cell-like structures. One famous study, led by chemist Bill Martin, showed how iron-sulfur minerals in vent walls could catalyze reactions resembling metabolic processes, essentially mimicking how living cells harvest energy. It's as if the vents handed life its first instruction manual. But here's the real kicker: these environments are remarkably stable. While Earth's surface was a chaotic mess of volcanic eruptions and radiation, the vents offered shelter. A consistent, mineral-rich oasis where delicate chemical processes could unfold uninterrupted for millennia. Some researchers argue this stability was crucial, giving those first fragile assemblies of molecules the time they needed to cross the threshold into life. Yet the hydrothermal vent hypothesis isn't without its mysteries. How did random chemistry become organized biology? Could these harsh, acidic environments really nurture life's delicate beginnings? And if life started here, why haven't we found fossil evidence in ancient seabed rocks? These questions keep scientists digging deeper, literally into one of the most provocative ideas about our origins. What we do know is this: if life did emerge from these scalding, mineral-laden waters, it would mean our earliest ancestor wasn't just hardy, it was a master of turning poison into possibility. Imagine a molecule so versatile it could store genetic information like DNA while also speeding up chemical reactions like a protein. That molecule isn't some futuristic invention, it's RNA, and it might have been the star player in life's origin story. This is the core of the RNA World hypothesis, a theory that suggests before DNA or proteins existed, tiny strands of RNA were the universe's first self-replicating machines. RNA is unique because it wears multiple hats. Unlike DNA, which is essentially a passive library of instructions, RNA can both carry genetic blueprints and fold into shapes that act as catalysts, molecular tools that drive chemical reactions. Some modern organisms still rely on RNA for critical functions, like ribosomes, the cellular factories that build proteins using RNA as both instruction manual and assembly line. Scientists realized if RNA could do this today, why couldn't it have been the starting point for life itself? Lab experiments have brought this idea to life. In the 1980s, researchers like Thomas Setch and Sidney Altman discovered ribozymes' RNA molecules that could cut and splice themselves without any protein help. Later, scientists like Jack Szostak engineered RNA strands that could copy themselves, albeit clumsily, under conditions mimicking early Earth. These findings were revolutionary. They showed RNA didn't just carry information, it could actively participate in its own reproduction, a crucial step toward biology emerging from chemistry. But the RNA World hypothesis isn't without its puzzles. For one, how did these fragile molecules survive long enough to replicate in a prebiotic world full of UV radiation and chaotic environments? RNA breaks down easily, especially without the protective enzymes modern cells provide. And while lab-created ribozymes can perform simple tasks, none yet replicate the complexity of even the simplest living systems. Some critics argue the leap from self-replicating RNA to a fully functional cell is like expecting a single sentence to rewrite itself into an entire book. Another sticking point is the origin of RNA itself. The building blocks of RNA nucleotides are complex molecules, and no one's entirely sure how they could have formed spontaneously in large enough quantities to kickstart life. Some researchers propose simpler molecules may have paved the way, while others suggest minerals or icy comets could have concentrated the necessary ingredients. Despite these gaps, the RNA World hypothesis remains one of the most compelling frameworks for understanding life's origins. It bridges the gap between chemistry and biology in a way few other theories can. If RNA truly was the first spark, it means life's earliest chapters weren't written in DNA's elegant double helix, but in RNA's messy, multitasking strands, a humble beginning for something as extraordinary as life itself. This theory also reshapes how we search for life beyond Earth. If RNA-based life emerged here, could it exist elsewhere? Missions to icy moons like Europa or Enceladus now look for environments where similar chemistry might unfold. The RNA world doesn't just explain our past, it might guide us toward discovering our cosmic neighbors. Yet the debate continues. Some scientists argue for a pre-RNA world, where even simpler molecules laid the groundwork. Others point to hybrid systems where RNA, proteins, and metabolism co-evolved from the start. What's remarkable isn't just the evidence we've gathered, but how much remains unknown. The RNA World hypothesis isn't a closed book. It's an open invitation to keep exploring, and that's the heart of science, not having all the answers, but asking better questions with each discovery. Clay might seem like the most ordinary stuff on Earth, just dirt beneath our feet. But what if this humble material played a starring role in the origin of life? Enter the clay catalysis theory, a fascinating idea suggesting that mineral surfaces, particularly certain types of clay, could have provided the perfect scaffolding for life's first molecules to assemble. Picture this: tiny organic compounds drifting through ancient pools of water, bumping into layers of clay. Instead of breaking apart, these molecules might have stuck to the clay's orderly surface, lining up like beads on a string. Over time, these organized gatherings could have grown more complex, eventually forming the building blocks of life. This theory owes much to the work of chemist Alexander Graham Cairns Smith, who proposed in the 1960s that clay minerals could have acted as a kind of molecular instruction manual. His insight was revolutionary. Clay isn't just a passive backdrop, it has a layered structure with repeating patterns, almost like a microscopic grid. These surfaces could have attracted and aligned simple organic molecules, guiding their interactions in ways that random collisions in water never could. Think of it as a molecular assembly line where clay's repeating crystal structure helped organize chaos into order. Experiments back this up. When scientists mix amino acids, the precursors to proteins, with certain clays, something remarkable happens. The clay doesn't just hold the molecules, it accelerates their bonding, encouraging them to link into longer chains. This process, called polymerization, is crucial for forming the complex molecules life depends on, like proteins and nucleic acids. In one striking study, researchers observed montmorillonite clay dramatically speeding up the formation of RNA-like strands. It's as if the clay provided a stage where chemistry could rehearse for biology. But why clay? For one, it was likely abundant on early Earth, especially around volcanic regions and tidal flats. Its charged surfaces could have acted like molecular Velcro, grabbing hold of organic compounds and holding them in place long enough to react. Some clays even contain metals like iron and zinc, which can act as catalysts, molecular matchmakers that lower the energy needed for reactions to occur. This combination of structure and chemistry makes clay a compelling candidate for life's first workshop. Yet the theory isn't without its skeptics. Some argue that while clay can assist in polymerization, it doesn't explain how these molecules gained the ability to replicate, a cornerstone of life. Others point out that not all clays behave the same way, and the right conditions might have been rare. Still, Cairn Smith's ideas opened a new way of thinking about life's origins, not just as a stroke of chemical luck, but as a process guided by the very geology of our planet. What's especially poetic about this theory is its simplicity. Life's grand beginnings might owe a debt to something as mundane as wet dirt. If clay truly played this role, it means the boundary between the living and non-living worlds isn't as sharp as we once thought. The same forces that shape mountains and canyons could have also shaped us, connecting the story of life to the story of Earth itself in a way that's both profound and strangely beautiful. This idea also raises broader questions. If clay can guide molecules toward complexity here, could it do the same elsewhere? Mars, for instance, has clay-rich regions, and missions like Curiosity have found organic molecules there. Could similar processes have unfolded on the Red Planet, even if they never progressed as far as they did on Earth? The clay catalysis theory doesn't just illuminate our past, it gives us a new lens for searching the cosmos. As with all origin theories, the clay hypothesis is a piece of a much larger puzzle. Maybe life didn't start in clay pools, but the evidence suggests they could have been one of many stages where chemistry inched closer to biology. What makes this idea so compelling isn't just the science, it's the reminder that sometimes the answers to life's biggest mysteries are hiding in plain sight, waiting in the most ordinary places. Long before scientists peered into hydrothermal vents or unraveled the secrets of RNA, there was a simpler idea, one that began with Charles Darwin himself. In a letter to a friend, he mused about a warm little pond where sunlight, heat, and the right chemicals might have conspired to create life. This poetic image became the foundation of the primordial soup theory, the notion that Earth's early oceans were a simmering broth of organic molecules, quietly brewing the first sparks of life. The theory took a giant leap forward in the 1950s when Stanley Miller and Harold Urey decided to put Darwin's pond to the test. Their now famous experiment sealed water, methane, ammonia, and hydrogen gases thought to dominate Earth's early atmosphere inside glass flasks. Then they fired sparks of electricity through the mix, simulating lightning strikes. Within days, the water turned pink, then murky brown. When Miller analyzed the sludge, he found amino acids, the very building blocks of proteins, swimming in the brew. It was a revelation. For the first time, science had shown that life's ingredients could arise from simple chemistry under conditions mimicking early Earth. But here's where the story gets even more interesting. Later discoveries revealed that Miller and Urey's original gas mix might not have matched Earth's true ancient atmosphere. Volcanoes, it turns out, likely pumped out carbon dioxide and nitrogen, not just methane and ammonia. Skeptics pounced. Did this invalidate the whole experiment? Surprisingly, no. When scientists revisited the experiment with updated volcanic gas blends, amino acids still formed, just different ones. Some even appeared more complex, hinting that volcanic activity could have supercharged the primordial soup rather than spoiled it. Another twist came from the oceans themselves. Early Earth seas weren't just passive bathtubs, they were dynamic theaters of chemistry. Tidal pools could have concentrated organic molecules through evaporation, like reducing a sauce to intensify its flavor. Hydrothermal vents, as we've seen, added heat and minerals to the mix. And then there's the role of sunlight. Near the ocean's surface, ultraviolet radiation might have broken apart simple molecules, only for them to reassemble into more complex forms, a destructive process that paradoxically fueled creation. Modern research has added yet another layer, the idea that Earth's primordial soup wasn't uniform. Instead, it might have been a patchwork of microenvironments, each with its own chemical quirks. A tide pool here, a volcanic seep there, a lightning-struck puddle somewhere else, each could have contributed unique ingredients to life's recipe. This patchwork view solves a key puzzle. While no single setting might have had everything needed for life, the combined efforts of many environments could have shuffled the deck until biology finally dealt itself a winning hand. Critics, of course, point out gaps. Amino acids are a start, but how did they organize into proteins? How did information storage, like RNA, enter the picture? And why haven't we found traces of this soup in ancient rocks? Yet the theory endures because it's more than just a lab experiment. It's a framework. It reminds us that life's origins might not hinge on a single eureka moment, but on countless small reactions playing out across millions of years in Earth's vast, roiling kitchen. What's striking is how Darwin's warm little pond still resonates today. The details have evolved, but the core idea, that life emerged from humble beginnings through the relentless work of chemistry, remains one of science's most enduring visions. And perhaps that's the most beautiful part, the notion that something as wondrous as life could arise from nothing more than water, gas, and time. Deep beneath the waves, where scalding water meets crushing pressure, another extraordinary theory takes shape, one where life's first sparks didn't just happen near hydrothermal vents but were literally forged in their fiery chemistry. Meet the iron-sulfur world hypothesis proposed by chemist Günther Wechtersheuser in the 1980s. His radical idea that life began not as a free-floating cell but as a chemical reaction clinging to the surface of pyrite fool's gold in the turbulent embrace of a hydrothermal vent. Wechtersheuser's vision flipped the script on traditional origin stories. Instead of life starting with replication or genetic material, he argued metabolism came first. Picture this: hot, mineral-rich fluid spewing from vents loaded with hydrogen sulfide and iron. When these chemicals mix, they form pyrite, releasing energy in the process. That energy, Wechtersheuser proposed, could have powered simple reactions, building blocks assembling into more complex molecules right on the pyrite surface. The mineral wouldn't just be a bystander. Its crystalline structure could have acted like a scaffold, organizing molecules into patterns that eventually became primitive metabolic pathways. The beauty of this theory lies in its simplicity. Pyrite's surface is positively charged, attracting negatively charged organic molecules like a magnet. Once stuck, these molecules could react with each other, driven by the vent's relentless energy. Over time, these reactions might have grown more sophisticated, forming cycles that resemble modern metabolism, like the citric acid cycle which powers our cells today. In Wechtersheuser's view, life didn't emerge from a soup but from a series of chemical handshakes on a microscopic metallic stage. Experiments lend credence to this idea. Scientists have recreated vent conditions showing how pyrite can catalyze reactions that produce acetic acid, a key player in metabolism. Other studies found that iron-sulfur clusters, similar to those in vents, sit at the heart of many modern enzymes, almost like molecular fossils of this ancient chemistry. It's as if life's earliest engines were built from spare parts left over from Earth's geologic infancy. Yet the iron-sulfur world isn't without challenges. Critics question how these surface-bound reactions could have transitioned into free living cells. And while pyrite can jumpstart chemistry, it's less clear how genetic information, like RNA, fit into this metabolic first scenario. Some scientists suggest a hybrid model where iron-sulfur chemistry and RNA cooperated from the start, each filling gaps in the other's story. What's undeniable is how this theory reshapes our imagination. Life might not have begun in a quiet pond or a gentle tide pool, but in the roaring, mineral-choked depths of an underwater forge. If Wechtersheuser is right, our earliest ancestor wasn't a cell, it was a chemical storm, clinging to a speck of pyrite, turning poison into power one reaction at a time. And that changes everything. If life can emerge from something so fierce and elemental, maybe it's not a rare miracle but an inevitable outcome of a planet with the right ingredients and enough time to stir them. This idea also echoes beyond Earth. Mars once had volcanic activity and possibly hydrothermal vents. Europa's icy shell hides a salty ocean warmed by similar forces. If iron and sulfur could midwife life here, why not there? The iron-sulfur world doesn't just explain origins, it gives us a new blueprint for where to look next. Hitchhiked theory reminds us that life's beginnings might have been less like a spark and more like a slow burn, a gradual alchemy of rock, water, and energy, writing the first lines of a story that would eventually include all of us. And in that story, pyrite isn't just a mineral, it's the universe's first battery, charging the chemistry that made life possible. What if life didn't actually begin anywhere because it's always been here? This is the spine-tingling premise of cosmic ancestry, a theory championed by astronomer Chandra Wickramasinghe. Unlike Panspermia, which suggests life traveled here from elsewhere, cosmic ancestry goes a step further. It proposes life is as eternal as the universe itself, woven into the fabric of space and time. Imagine microbes not as hitchhikers on comets but as cosmic constants, as inherent to existence as stardust or gravity. Wickramasinghe's ideas build on decades of collaboration with Fred Hoyle, the same scientist who championed Panspermia. Together, they argued that the universe is teeming with complex organic molecules, and possibly even dormant life scattered across galaxies. But cosmic ancestry takes this further by rejecting the idea of abiogenesis entirely. Instead of life emerging from non-life on Earth or elsewhere, Wickramasinghe suggests life has always existed, riding comets and interstellar dust to new worlds like Earth. It's a theory that blurs the line between biology and cosmology, framing life as a fundamental feature of reality. The evidence Wickramasinghe points to is as intriguing as it is contentious. Interstellar dust clouds, once thought to be barren, are now known to harbor complex organics like formaldehyde and ethanol. Meteorites, like the infamous Murchison meteorite, carry not just amino acids but nucleobases, the letters of DNA's alphabet. In 2008, scientists even detected a faint but unmistakable signature of organic material in the light from distant galaxies, suggesting these molecules might be universal. For cosmic ancestry proponents, this isn't just coincidence, it's proof that life's ingredients are baked into the cosmos, yet mainstream science remains skeptical. Critics argue that finding organic molecules isn't the same as finding life. Just because the universe provides the building blocks doesn't mean it's always building. There's also the question of how delicate microbial life could survive billions of years in the vacuum of space, exposed to radiation and extreme temperatures. Even the hardiest extremophiles have limits, and then there's the philosophical hurdle. If life didn't start anywhere, how do we explain its complexity? Cosmic ancestry sidesteps the origin question entirely, which some scientists feel is less a solution than a surrender. But Wickramasinghe's theory forces us to think bigger. If life is truly a cosmic inevitability, then every planet, moon, or rogue comet could be a potential cradle. It reshapes the search for extraterrestrial life from hunting for evidence to expecting ubiquity. And it challenges our very definition of life, suggesting it might not be a rare fluke but a universal law, as intrinsic to the universe as light or gravity. Whether cosmic ancestry ever gains widespread acceptance, it serves as a thrilling counterpoint to Earth-centric origin stories. It dares us to imagine a universe where life isn't the exception but the rule, a whisper in the cosmic wind that's always been there, waiting for the right conditions to speak aloud. And in that vision, Earth isn't life's starting point, it's just another stage in an eternal performance. Forget the idea of cells evolving late in life's origin story. What if they came first? That's the bold claim of the lipid world hypothesis, a theory that flips the script by suggesting life didn't start with genetic material or metabolism, but with bubbles. Not just any bubbles, though tiny, self-assembling spheres of lipids, the same fatty molecules that make up cell membranes today. Picture a shallow pool on early Earth where waves or rain create foamy slicks on the surface. In this froth, lipid molecules could have spontaneously organized into hollow vesicles, trapping pockets of water inside like microscopic balloons. These weren't cells yet, but they might have been the universe's first containers, setting the stage for everything that followed. The magic of lipids lies in their chemistry. Each molecule has a head that loves water and a tail that hates it, a duality that drives them to arrange themselves into double-layered sheets, curling into sealed spheres when shaken in water. In labs, this happens effortlessly. Mix the right lipids under conditions mimicking early Earth, and within minutes you'll see vesicles forming, some even growing, dividing, and merging on their own. Remarkably, these structures can trap other molecules inside, creating isolated microenvironments where chemical reactions could concentrate and interact without diluting away. It's like nature invented tiny test tubes before it figured out the experiments. But here's where the theory gets truly exciting. These lipid vesicles aren't just passive bags, they're dynamic. Experiments show they can undergo selection pressure, much like living organisms. Vesicles with more stable membranes persist longer, while fragile ones burst, leaving behind their content. Add in the fact that certain lipids can act as rudimentary catalysts, and suddenly, these bubbles aren't just containers, they're participants in the chemical drama, actively shaping which reactions succeed or fail. Some researchers have even coaxed vesicles to steal lipids from each other, competing for resources in a primitive mimicry of cellular competition. The implications are profound. If lipid vesicles emerged early, they could have provided the physical framework for genetics and metabolism to later co-opt. Imagine RNA strands or iron-sulfur clusters becoming trapped inside these bubbles, suddenly shielded from the chaos of the outside world. Over time, the most successful combinations, vesicles housing molecules that enhanced stability or reproduction, would have outlasted the rest, nudging chemistry closer to biology. Critics, of course, point to gaps. How did these simple sacs transition into true cells with programmable heredity? And why haven't we found fossilized lipids in ancient rocks? Yet the lipid world offers something rare in origin of life research, a tangible bridge between the non-living and the living. These vesicles don't require sophisticated chemistry to form, just the right ingredients and a little agitation, both of which early Earth had in abundance. Perhaps the most poetic aspect of this theory is how it mirrors life's essence. Cells, after all, are defined by their boundaries, the membranes that separate self from non-self. If the lipid world is correct, then life's first act wasn't metabolism or replication, but something far simpler, drawing a line in the water and declaring, "This inside is different from the outside." From that humble division, all else may have flowed. This idea also reshapes how we search for life beyond Earth. If lipid vesicles can form easily, then icy moons with subsurface oceans or even methane lakes on Titan could harbor similar structures. We might not find DNA out there, but we could stumble upon bubbles quietly doing what they've done for billions of years, testing the boundaries between chemistry and life, one microscopic sphere at a time. Quantum mechanics and the origin of life, two ideas that seem worlds apart, until you peer closely at the strange dance of particles inside every living cell. This is the radical premise of the quantum life hypothesis, that the spooky, probabilistic rules governing atoms and electrons didn't just influence life, they might have helped create it. At the intersection of physics and biology, thinkers like Paul Davies have proposed that quantum phenomena like entanglement, tunneling, and coherence could have provided the missing spark that transformed chemistry into biology. The theory hinges on a startling observation. Life's core processes rely on staggeringly precise molecular interactions. Enzymes, for instance, speed up reactions by factors of trillions, a feat physicists once thought impossible without quantum tunneling, where particles teleport through energy barriers they classically couldn't cross. Photosynthesis, too, shows signs of quantum weirdness. Experiments reveal that energy in plant cells doesn't hop randomly between molecules. It explores multiple pathways simultaneously, like a wave testing all possible routes before collapsing into the most efficient one. This quantum coherence, once thought too fragile for warm, wet biology, appears to be a hidden tool in nature's workshop. Davies and others argue that if quantum effects optimize life today, perhaps they also guided its beginnings. Picture a probiotic molecule on early Earth, vibrating between different states. Quantum superposition, the same phenomenon that lets Schrödinger's cat be both dead and alive, might have allowed molecules to test multiple chemical configurations at once, dramatically speeding up the search for stable, self-replicating structures. In this view, life didn't emerge despite quantum physics. It emerged because of it, leveraging the universe's built-in shortcuts to navigate the vast combinatorial desert of possible molecules. Skeptics, however, raise eyebrows. Biologists point out that while quantum effects exist in living systems, they're carefully orchestrated by complex cellular machinery. How could such delicate phenomena survive the noisy, disordered environment of early Earth? Critics also note that demonstrating quantum effects in modern biology doesn't prove they enabled abiogenesis. It might be a case of life later harnessing quantum tricks, not being born from them. Yet the quantum life hypothesis refuses to fade, fueled by discoveries like quantum vibrations in DNA and evidence that bird navigation relies on entangled electrons. Whether it ultimately solves the mystery or not, this theory stretches our imagination, suggesting life's origins might be tied to the deepest, most counterintuitive layer of reality. After all, if quantum physics can birth stars and shape atoms, why not life itself? The answer, as always in this grand puzzle, remains tantalizingly just out of reach. Volcanic glass or obsidian might seem like an unlikely candidate for life's origin story after all. It's just shiny black rock formed from cooling lava. But peer closer at its jagged edges under a microscope, and you'll find a hidden world of tiny pores and tunnels, a labyrinth that could have served as nature's earliest laboratory. This is the volcanic glass catalysis theory, which suggests that the razor-sharp surfaces and microscopic chambers of obsidian might have provided the perfect stage for life's first chemical performances. The magic lies in obsidian's structure. When lava cools rapidly, it traps gas bubbles, creating a porous network riddled with nooks and crannies. These cavities aren't just empty space. They're potential reaction vessels, each one acting like a miniature flask where organic molecules could gather, shielded from the chaos of the outside world. Even better, volcanic glass carries a slight electric charge, thanks to the way its silicon and oxygen atoms align. This charge could have attracted and stabilized delicate molecules, luring them into close proximity where they might react. It's like the rock itself was a magnet for chemistry, pulling the pieces of life's puzzle together before biology even knew how to solve it. Experiments back up this idea brilliantly. In a landmark study, scientists simulated early Earth conditions by mixing simple organic compounds with powdered obsidian. The results were striking. The volcanic glass didn't just passively host reactions, it actively accelerated them. Amino acids linked up faster, forming longer chains than they did in solution alone. Some researchers even observed the formation of protomembranes, delicate bubbles resembling cell walls clinging to the glass's jagged surfaces. These findings hint that obsidian's pores didn't just concentrate molecules, they might have helped organize them into more complex structures, nudging chemistry toward biology one tiny cavity at a time. What makes this theory especially compelling is its simplicity. Obsidian was everywhere on early Earth, a byproduct of the planet's fiery volcanic youth. Tidal pools or hot springs lined with volcanic glass would have been natural reactors, their pores teeming with concentrated organic soups. And unlike some other proposed catalysts, obsidian doesn't require rare metals or extreme temperatures, just water, time, and the right molecules. This universality suggests that if life did start this way, it might not have been a fluke, but an inevitability wherever volcanic activity met liquid water. But the volcanic glass theory isn't without its mysteries. How did these surface-bound reactions transition into free-floating cells, and why haven't we found direct evidence of this process in ancient rocks? Some critics argue that while obsidian can facilitate reactions, it doesn't explain how information storage like RNA entered the picture. Yet even skeptics admit the theory's elegance. It turns a commonplace geologic material into a potential architect of life, a humble rock with a hidden talent for chemistry. This idea also reshapes how we search for life beyond Earth. Mars, for instance, boasts vast fields of ancient volcanic glass. If these rocks could have midwifed life here, why not there? Future missions might prioritize sampling these glittering formations, hunting for molecular fossils trapped in their glassy embrace. The volcanic glass hypothesis reminds us that life's beginnings might have been less about exotic environments and more about ordinary rocks doing extraordinary things, one tiny pore at a time. Beneath our feet, miles below the ocean floors and mountain ranges, lies a world as alien as any distant planet, a realm where life might have taken its very first breath. This is the deep Earth hypothesis, a theory proposing that life didn't emerge in sunlit ponds or hydrothermal vents, but in the crushing darkness of the planet's crust, powered not by sunlight or chemical soups, but by the slow, steady decay of radioactive rocks. Picture a labyrinth of microscopic cracks and fissures where water seeps through granite and basalt, warmed by the heat of uranium, thorium, and potassium. In these hidden chambers, shielded from the violent surface conditions of early Earth, simple molecules could have found the time and energy to assemble into something far more extraordinary. The theory gained traction with the discovery of the deep subsurface biosphere, a vast ecosystem of microbes thriving kilometers below ground, far beyond the reach of sunlight. These organisms, dubbed intraterrestrials, survive on chemosynthesis, just like their hydrothermal vent cousins, but with a twist. They harness energy not from sulfur or methane, but from hydrogen gas produced when water reacts with iron-rich minerals. Even more astonishing, some species feed directly on radiation, using the energy from radioactive decay to power their metabolism. If microbes can thrive in such extreme conditions today, why couldn't life have started there billions of years ago? Supporters of this idea point to the unique advantages of the deep crust. Unlike the surface, which was bombarded by asteroids and scorched by ultraviolet radiation, the subsurface offered a stable, shielded environment. Radioactive minerals like uraninite would have provided a steady trickle of energy over millions of years, acting as natural nuclear reactors for early chemistry. Lab experiments back this up. When scientists simulate deep Earth conditions mixing water, minerals, and radiation, they observe the formation of organic molecules, even primitive cell-like structures. It's as if the rocks themselves were quietly conducting experiments, waiting for the right combination to spark life. But the deep Earth hypothesis isn't without its challenges. Critics argue that while radiation can drive reactions, it also breaks down complex molecules, a delicate balance that might have hindered rather than helped life's origins. Others question how subsurface life could have migrated upward to colonize the surface, or why we haven't found clearer fossil evidence in ancient rocks. Yet the theory's allure lies in its universality. If life can emerge in the deep crust, then any rocky planet with water and radioactive decay could, in theory, harbor similar beginnings. Mars, for instance, once had active geology and possibly even subsurface water. Could its depths still hold echoes of a life that never made it to the surface? Perhaps the most profound implication is what this says about life's resilience. The deep Earth isn't just a niche, it's a vast, unexplored frontier, making up most of the planet's habitable volume. If life did start there, it means our search for origins shouldn't just look up to the stars, but down into the very bones of our world, and that changes everything. The story of life might not be written in ancient oceans or cosmic dust, but in the quiet, glowing heart of the planet itself, a testament to the fact that life, once it begins, finds a way to endure even in the unlikeliest of places. Now imagine the edge of an ancient shoreline where the rhythm of the tides creates a natural laboratory unlike any other, a place where water pulses in and out, leaving behind concentrated pools teeming with possibility. This is the tidal pool theory, which suggests that life's first molecules didn't just form in water but thrived because of its absence, relying on the cycles of wetting and drying to drive chemistry forward. Picture a shallow basin carved into volcanic rock, filled with organic, rich broth as the tide rolls in. Then, under the sun's glare, the water evaporates, leaving behind a thick, salty residue where molecules jostle closer than ever before. In that concentrated chaos, bonds form, chains lengthen, and complexity emerges not by chance, but by the relentless push and pull of the ocean's heartbeat. The genius of this theory lies in its simplicity. Tidal pools aren't exotic or rare, they're universal features of coastlines both then and now, but their power comes from repetition. Each cycle of flooding and evaporation acts like a molecular sieve, filtering out weaker interactions and amplifying the strong ones. Research backs this up spectacularly. In labs, scientists have simulated tidal pool conditions by repeatedly wetting and drying mixtures of amino acids. The results, peptide short chains of amino acids, form at rates hundreds of times faster than in static solutions. Some experiments even produce protein-like structures, hinting that these pools could have been nature's first assembly lines, turning simple ingredients into the building blocks of life one tide at a time. What makes tidal pools especially compelling is their dynamic range. When wet, they're a soup of dissolved molecules. When dry, they become microscopic reaction chambers where proximity sparks chemistry. Add in factors like temperature swings, mineral catalysts from surrounding rocks, and even ultraviolet light breaking and remaking bonds, and you have a recipe for complexity. It's as if the tides themselves were stirring the pot, guiding randomness toward order. But the theory doesn't stop at molecules. Tidal pools also solve a puzzle other models struggle with: how early life might have transitioned from chemistry to compartmentalization. As water evaporates, lipids and other hydrophobic molecules can self-organize into membranous layers, creating natural enclosures. These protocells, forged in the ebb and flow, would have had ready-made boundaries, protecting their contents while still allowing exchange with the environment. In essence, tidal pools don't just build molecules, they package them into something resembling life's first homes. Critics, of course, raise valid questions. Would the harsh UV light of early Earth have destroyed delicate polymers faster than tides could create them? Could random tidal fluctuations reliably concentrate the right ingredients? Yet the theory's elegance is hard to dismiss. Unlike deep-sea vents or volcanic chambers, tidal pools offer a bridge between land and sea, a testing ground where molecules could experiment with both aquatic and terrestrial chemistries. This idea also reshapes how we view life's resilience. If tidal pools were life's cradle, then the boundary between land and ocean isn't just a geographic line, it's a creative frontier. And that insight extends beyond Earth. Mars once had vast shorelines, and Saturn's moon Titan boasts methane tides. If tidal dynamics birthed life here, why not in other worlds with rhythmic liquid cycles? The tidal pool theory isn't just about origins. It's a reminder that life might flourish wherever there's a steady pulse, a place where the universe can whisper its secrets between the rise and fall of the waves. The universe has a funny way of delivering gifts, sometimes wrapped in fire and hurtling through the atmosphere at tens of thousands of miles per hour. That's the essence of the meteorite delivery theory, which suggests Earth's early chemistry might have gotten a cosmic boost from space rocks packed with organic molecules. Take the Murchison meteorite, which lit up the Australian sky in 1969. When scientists cracked open its charcoal-black exterior, they didn't just find ordinary rock. They discovered a treasure trove of amino acids, the very building blocks of proteins. Not just one or two varieties either, over 90 different types, including some not commonly found on Earth. It was as if the cosmos had sent a care package stuffed with life's potential ingredients. But amino acids were just the beginning. Later analyses revealed even more stunning finds: nucleobases, the molecular letters that spell out DNA and RNA. Pyrimidines and purines like uracil and xanthine, compounds essential for genetic coding, were nestled in the meteorite's ancient matrix. Even more astonishing, some of these molecules showed a slight excess of left-handed chirality, a mirror image preference also seen in Earth's biology. Coincidence or a hint that life's asymmetry might have interstellar roots? The implications are staggering. If meteorites like Murchison were common in Earth's youth, they could have carpet-bombed the planet with organics, turning our oceans into probiotic soups with extraterrestrial seasoning. Calculations suggest that during the late heavy bombardment around 4 billion years ago, Earth may have received thousands of tons of organic material annually from comets and asteroids. That's not just a sprinkle of ingredients, it's a full cosmic pantry. Yet this theory isn't about finding alien microbes hitchhiking on space rocks. It's subtler and more scientifically plausible. The idea isn't that life itself arrived from space, but that the raw materials for life did, giving Earth's chemistry a head start. Think of it like receiving a box of high-quality kitchen ingredients versus a pre-cooked meal. The meteorites delivered the flour, sugar, and eggs, not the cake. Experiments back this up. When scientists simulate the conditions of interstellar space, ultra-cold vacuums peppered with ultraviolet radiation, they can create complex organics like amino acids and sugars on icy dust grains. These molecules then hitch rides on comets and asteroids, surviving the journey through space thanks to the protective embrace of rock and ice. Some even endure atmospheric entry, shielded inside the meteorite's core. Critics, however, raise valid questions. Could these delicate molecules really have survived Earth's violent early environment? And if space was such a prolific chemistry lab, why haven't we found more evidence of these processes in younger meteorites? Despite these gaps, the meteorite theory reshapes how we see our place in the cosmos. Earth wasn't an isolated incubator. It was part of a vast, interconnected chemical network, with comets and asteroids as its delivery vehicles. This idea also transforms the search for life beyond Earth. If organics rain down everywhere, then planets and moons across the galaxy might have received similar starter kits. Europa's icy crust, Mars' ancient lakebeds, even Titan's methane lakes, all could harbor molecules forged in the depths of space. The meteorite theory doesn't just explain our origins, it suggests the universe might be quietly, relentlessly, seeding the possibility of life wherever it can. So the next time you see a shooting star, remember, it might be more than a fleeting light. It could be a remnant of the ancient cosmic courier service that helped write the first lines of life's story. And that story, it seems, was always meant to be written in the stars. Sunlight might seem too harsh to nurture life's delicate beginnings, but what if it was the very spark that set everything in motion? This is the heart of the photochemical origins theory, the idea that ultraviolet light streaming through Earth's early atmosphere acted like a cosmic chemist, stitching together simple molecules into the complex ingredients of life. Picture our planet 4 billion years ago, bathed in relentless UV radiation, with no ozone layer to soften the blow. Instead of sterilizing the surface, this barrage of energy could have supercharged shallow pools and tidal flats, turning them into nature's oldest laboratories. The evidence for this comes from stunningly simple experiments. In the 1950s, biochemist Juan Oro recreated early Earth conditions by shining UV light onto a mixture of water, ammonia, and hydrogen cyanide gases thought to be abundant back then. Within days, the solution teamed with adenine, one of the 4 nucleobases that form DNA. It was a revelation. Sunlight alone could assemble life's alphabet from scratch. Later studies expanded on this, showing UV light could also synthesize sugars, amino acids, and even the precursors to lipids. Suddenly, the same radiation we slather on sunscreen to block became a prime suspect in life's origin story. But how does it work? UV photons pack enough energy to break chemical bonds, but destruction isn't the only outcome. When these high-energy rays hit molecules like hydrogen cyanide or formaldehyde, they can trigger cascading reactions, forging new bonds in the wreckage. Think of it as molecular origami. The light crumples simple compounds, and when they unfold, they've transformed into something more intricate. Shallow pools would have been ideal for this process, concentrating molecules during dry phases while UV light worked its magic. Over countless cycles, these reactions could have stockpiled life's raw materials, like a cosmic assembly line running on sunlight. One of the most compelling pieces of this puzzle comes from a surprising place: modern meteorites. Scientists found that certain carbon-rich asteroids contain molecules called quinons, which excel at absorbing UV light and using its energy to drive chemical reactions. If asteroids carried these photochemical catalysts to early Earth, they might have turbocharged the process, acting like molecular solar panels to harness sunlight for building complexity. It's a tantalizing link between space chemistry and terrestrial biology, one that suggests the universe might have handed life a ready-made toolkit. Critics, of course, point to the elephant in the room. UV light doesn't just create, it destroys. Expose delicate RNA or proteins to unfiltered sunlight, and they degrade rapidly. But here's where nature's ingenuity shines. Mineral surfaces like clay or titanium dioxide could have shielded newly formed molecules, while tidal rhythms might have allowed daytime synthesis followed by nighttime protection in deeper waters. Some researchers even propose that sulfur compounds in early oceans acted as a natural sunscreen, filtering out the most damaging wavelengths while letting through just enough energy to power chemistry. The photochemical theory doesn't just rewrite life's origin story, it reshapes how we search for it elsewhere. Europa's icy shell, for instance, cracks under Jupiter's radiation, potentially creating UV-driven chemistry in subsurface pools. Mars' ancient clay deposits might have harbored similar reactions when the sun was younger and more violent. If light alone can jumpstart life's ingredients, then the universe's recipe book just got a lot simpler. What's poetic about this idea is its duality. The same force that threatens life today might have been its midwife. It's a reminder that creation and destruction often share the same source, and that life, against all odds, found a way to rise from the chaos. The photochemical theory doesn't just answer how life began, it whispers a deeper truth. Sometimes you need the storm before the seed can sprout. Deep beneath the waves, where scalding mineral-rich fluids seep from the seafloor, lies a theory that could rewrite our understanding of life's first energy source. The alkaline vent hypothesis, championed by geochemist Mike Russell, proposes something extraordinary: that the very same chemical trick modern cells used to power themselves might have been invented not by biology, but by geology, billions of years before the first true cell even existed. Picture a towering hydrothermal vent, not the black smoker type we've seen before, but a gentler, more labyrinthine structure known as a white smoker. Its porous, honeycombed walls, made of minerals like iron and sulfur, create a natural electrochemical reactor where something magical happens. Proton gradients form spontaneously, mirroring the energy-generating process inside every living cell today. Here's why this matters. All life as we know it runs on proton gradients. Your mitochondria, the power plants of your cells, pump protons across membranes to create an imbalance, a sort of microscopic waterfall. When those protons flow back down, their energy is harnessed to build ATP, the universal molecular battery. Russell's insight was that alkaline vents could have done this first, using chemistry instead of biology. The vents' warm, alkaline fluids rich in hydrogen meeting the cooler, acidic ocean water loaded with carbon dioxide create a natural voltage difference, a proto-battery built into Earth itself. Minerals in the vent walls act like primordial membranes, separating these fluids and maintaining the gradient, just as cell membranes do today. It's as if the planet sketched the blueprint for metabolism before life even knew how to read it. Experiments bring this idea to life. When scientists recreate vent conditions in labs, they observe the formation of tiny metal sulfide bubbles, their walls laced with microscopic pores. These bubbles, much like cell membranes, can trap molecules inside while maintaining chemical imbalances across their walls. Some even generate voltage, mimicking the energy production of living cells. The kicker, these structures self-assemble without any biological input, just chemistry obeying the laws of physics. Russell's team showed that such vents could have produced not just energy, but also organic molecules. Hydrogen from the vents reacts with carbon dioxide in seawater, forming intermediates like formate and acetate, key players in modern metabolism. Suddenly, the line between geology and biology blurs. This theory also offers a compelling answer to one of abiogenesis' biggest puzzles: how life standardized its energy currency so early. Luca, the last universal common ancestor of all living things, already had sophisticated proton-driven metabolism. If vents provided this mechanism ready-made, it explains why all life inherited the same system, not through convergent evolution, but because it was the only game in town. The vents didn't just supply energy, they handed life its first operating manual. Critics, of course, question whether these mineral gradients could have been stable enough to sustain proto-life, or how the system transitioned from rock-bound chemistry to free-floating cells. Yet the alkaline vent hypothesis has an elegance that's hard to ignore. It turns life's most universal process into a planetary inheritance, a gift from Earth's own geochemistry. And if this is how life began here, then similar vents on icy moons like Enceladus or Europa could be running the same ancient software, silently waiting for biology to hit start. What's haunting about this idea is its implication that the spark of life might not have been a spark at all, but a slow, steady hum of energy whispering through mineral walls for eons before the first cell ever stirred. And in that whisper, the universe was quietly teaching chemistry how to become something more. Long before plants mastered the art of turning sunlight into sugar, Earth's earliest life forms might have dined on something far more pungent: sulfur. This is the sulfur metabolism first theory, a compelling argument that life's first energy source wasn't the golden rays of the sun, but the stinky, reactive chemistry of sulfur compounds bubbling up from geothermal vents and volcanic pools. Picture a world without oxygen, where the air reeked of rotten eggs and the oceans shimmered with milky clouds of sulfur. In this hostile environment, the ability to harness sulfur's volatile energy could have been the difference between survival and oblivion for our planet's pioneering organisms. The evidence for this idea runs deep, literally. Some of Earth's most ancient microbes, like the heat-loving archaea found in Yellowstone's boiling springs, still rely on sulfur metabolism today. These extremophiles thrive in conditions resembling early Earth, reducing sulfur compounds to generate energy in a process eerily similar to how we use oxygen. Even more telling, when scientists analyze the oldest sedimentary rocks, they find layers rich in pyrite fool's gold, a mineral that forms when microbes process sulfur. It's as if the planet itself left fingerprints of this primordial metabolism in the geologic record. But why sulfur? The answer lies in its chemistry. Sulfur exists in multiple states, easily shuttling between forms like sulfide, sulfite, and sulfate. This versatility makes it a perfect biological battery able to accept and donate electrons with minimal effort. In the oxygen-free hellscape of early Earth, sulfur reactions would have been one of the few reliable energy sources, especially near hydrothermal vents where hydrogen sulfide spewed from the seafloor. Some researchers argue these environments were nature's first power plants, with mineral catalyzed sulfur reactions providing a template for later metabolic pathways. Experiments back this up spectacularly. When scientists simulate vent conditions with iron, sulfur, and carbon dioxide, they observe the spontaneous formation of simple organic molecules, with sulfur acting as both catalyst and fuel. Even more striking, certain modern enzymes at the heart of sulfur metabolism contain iron-sulfur clusters, molecular artifacts that might be relics of this ancient chemistry. It's like finding a fossil, but instead of bones, it's a microscopic machine still humming along in our cells today. This theory also reshapes how we view life's earliest innovations. Before photosynthesis revolutionized the planet, sulfur metabolism could have sustained entire ecosystems in the deep ocean's darkness, much like the lush microbial mats around today's black smokers. And because sulfur cycles don't require sunlight, this opens the possibility that life could arise in similarly sulfur-rich environments elsewhere, like the subsurface oceans of Europa or the acidic clouds of Venus. Yet the sulfur world isn't without its mysteries. How did early organisms transition from relying on ambient sulfur chemistry to actively managing it? Why don't all modern life forms retain sulfur metabolism if it was so foundational? These gaps keep scientists digging, sometimes literally, into rocks and extremophiles for clues. What's undeniable is sulfur's enduring legacy. Every time you taste the metallic tang of a mineral spring or catch a whiff of a volcanic hot spring, you're smelling the remnants of a world where sulfur wasn't just a nuisance, it was the spark that kept life's flame alive in a young, chaotic universe. Ice might seem too harsh and lifeless to cradle the first sparks of biology, but what if its very structure held the key to organizing chaos into order? This is the icy crucible theory, the idea that freezing temperatures didn't stifle life's origins but accelerated them, using ice's crystalline lattice as a molecular scaffold. Picture Earth 4 billion years ago, its shallow oceans periodically freezing under a faint young sun. Within those ice sheets, microscopic veins of liquid brine would have trapped pockets of organic molecules, squeezing them into close quarters while shielding them from destructive ultraviolet radiation. In this frigid laboratory, the sluggish pace of cold chemistry might have been life's unlikely ally, allowing fragile compounds to stabilize, interact, and ultimately replicate in ways impossible in warmer, more chaotic environments. Experiments reveal ice's surprising talent for fostering complexity. When scientists simulate probiotic conditions by freezing mixtures of water, ammonia, and simple organic compounds, something remarkable happens. The freezing process concentrates impurities, including potential building blocks of life, into microscopic liquid veins between ice crystals. These crowded pockets become hubs of chemical activity, with molecules colliding far more frequently than in open water. Even more striking, ice's crystalline structure can align molecules in precise orientations, almost like a molecular assembly line. In one groundbreaking study, researchers observed RNA strands, life's potential first genetic material, replicating more efficiently in ice than in liquid water. The cold didn't hinder the process, it organized it, providing a stable framework where fragile reactions could unfold without falling apart. The secret lies in ice's dual nature. While solid ice appears static, its microscopic environment is anything but. Freeze-thaw cycles like those on early Earth's tidal flats or glacial ponds would have repeatedly concentrated and mixed organic compounds, creating pulses of chemical activity. Meanwhile, ice's UV-absorbing properties could have protected delicate molecules from the sun's sterilizing rays, offering a natural sunscreen for nascent biology. Some researchers even suggest ice's hydrogen-bonded structure mimicked the scaffolding of modern cell membranes, providing primitive compartments where metabolism and genetics could co-evolve. This theory gains extra weight when we look beyond Earth. Comets, those icy relics of the solar system's birth, are packed with organic molecules. Europa and Enceladus, moons with subsurface oceans beneath icy shells, show signs of hydrothermal activity, raising the possibility that similar ice-bound chemistry could be unfolding there right now. If life could start in Earth's frozen slush, why not in similar environments across the cosmos? Critics, of course, question whether ice-based reactions could have generated enough complexity to leap toward true biology. Yet the icy crucible hypothesis offers something rare, a bridge between the destructive chaos of early Earth and the delicate order life requires. Ice wasn't just a passive backdrop, it was an active participant, shaping chemistry with the quiet precision of a master craftsman. And in that paradox, life emerging from what seems like a frozen desert lies a profound lesson. Sometimes the most barren places hold the seeds of the extraordinary. Out in the vast emptiness between stars, where temperatures hover just above absolute zero and atoms drift alone for millennia, something extraordinary happens. Cosmic dust becomes a cradle for chemistry. The cosmic dust assembly theory flips our earthly perspective, suggesting the first building blocks of life didn't just form on a planet but were pieced together in the frigid void of space, on the surface of tiny mineral grains no wider than a human hair. These microscopic motes, forged in the hearts of dying stars and scattered across galaxies, might have served as the universe's first workbenches, providing the perfect surface for simple molecules to meet, bond, and grow into something far more complex. The magic begins in stellar nurseries, dense clouds of gas and dust where new stars are born. Here, shielded from harsh radiation in the icy darkness, something remarkable unfolds. Atoms of hydrogen, carbon, and oxygen stick to dust grains like frost on a windowpane, forming icy mantles. But these aren't just frozen water. Over eons, cosmic rays and ultraviolet light bombard these ices, breaking and rearranging their bonds into more elaborate structures. Lab experiments simulating these conditions reveal a stunning truth. When exposed to space-like radiation, simple ices transform into organic compounds like sugars, amino acids, and even the nucleobases that make up RNA. It's as if the universe has a hidden recipe written in starlight and dust for life's ingredients. Astronomers have spotted evidence of this process across the cosmos. The Orion Nebula, a stellar nursery visible to the naked eye, contains vast quantities of methanol and formaldehyde. The Taurus molecular cloud holds glycolaldehyde, a simple sugar. Even more complex molecules like ethyl formate, which gives raspberries their scent, drift through Sagittarius B2, a massive gas cloud near the Milky Way's center. These discoveries suggest interstellar space isn't just a barren wasteland but a sprawling chemical factory, quietly assembling the pieces of life one dust grain at a time. But how did these cosmic creations reach Earth? The answer lies in comets and meteorites, celestial couriers that transport preserved stardust across the solar system. When our planet was young, it was bombarded by these icy visitors, delivering their cargo of pre-made organic molecules directly to Earth's surface. The Murchison meteorite, for instance, contained over 90 amino acids, many not commonly found on our planet. Similarly, the Rosetta mission detected glycine, the simplest amino acid, in the coma of Comet 67P. These findings hint that Earth's primordial soup might have been seasoned with ingredients brewed in deep space. What makes this theory so compelling is its universality. The same processes unfolding in our solar system's infancy could be happening right now in distant star systems. If cosmic dust acts as a molecular workshop everywhere, then the raw materials for life might be scattered throughout the galaxy, waiting for the right planet to land on. It's a humbling thought, the idea that life's beginnings weren't just an earthbound accident but part of a grand cosmic assembly line, with stardust as its silent craftsman. Yet mysteries remain. Can delicate molecules survive the brutal heat of atmospheric entry? How did space-borne organics integrate with Earth's chemistry to spark biology? These questions keep scientists probing meteorites and designing lab experiments that mimic the cold vastness of space. But one thing is clear: the universe seems pre-wired for chemistry, with dust grains as its unsung architects. This idea also reshapes how we search for life beyond Earth. Missions like NASA OSIRIS-REx, which returned samples from the asteroid Bennu, aren't just collecting rocks. They're reading a chemical love letter from the cosmos, one written in the language of dust and ice. And in that message, there's a tantalizing suggestion. Life's story might not begin on planets at all, but in the glittering darkness between them, where the smallest specks hold the biggest secrets. Imagine a world before genes, before cells, before even the simplest forms of replication, yet somehow, chemistry still found a way to organize itself into something resembling life. This is the heart of Stuart Kauffman's autocatalytic set theory, a radical idea that challenges one of biology's deepest assumptions, that replication had to come first. Instead, Kauffman proposed that life's origins might have begun with something far more subtle, a tangled web of chemical reactions where molecules didn't just interact, but actively produced the very substances that kept the whole network alive. Picture a candle flame, not because it replicates, but because it sustains itself through a cascade of reactions. What if life started not with a single replicator, but with an entire orchestra of molecules playing in perfect, self-renewing harmony? The genius of autocatalytic sets lies in their simplicity. Take a pool of diverse organic molecules, amino acids, sugars, lipids, and let them interact randomly. Most reactions fizzle out, but occasionally something extraordinary happens. One molecule catalyzes the formation of another, which in turn helps create a third, and eventually some of these products circle back to jumpstart the original reaction. This closed loop is the essence of an autocatalytic set, a system that feeds itself, growing more complex not through genetic instructions, but through the sheer inevitability of chemistry. Kauffman's mathematical models show that in a sufficiently diverse molecular soup, such self-sustaining networks aren't just possible, they're inevitable. Experiments bring this theory to life. In a lab, scientists have recreated primitive autocatalytic cycles using nothing more than simple peptides and RNA fragments. One striking example involves the Formose reaction, where formaldehyde molecules link up into sugars without any enzymes, just a push from a few mineral catalysts. What starts as a disorganized mix soon settles into a rhythmic dance of creation and renewal, with each sugar molecule aiding the formation of the next. It's not replication in the traditional sense, but it's something just as powerful. A system that maintains itself against the pull of entropy, the universe's relentless slide toward disorder. This shifts how we view life's earliest moments. Instead of a single eureka molecule like RNA struggling to copy itself, autocatalytic sets suggest a messier, more collaborative origin, one where complexity emerged from the collective effort of many molecules, each propping up the others. Modern cells still echo this ancient teamwork. Metabolic pathways like the Krebs cycle are essentially autocatalytic, with each step producing molecules that fuel the next. Even our ribosomes, the protein-building machines, rely on a dance of RNA and proteins that some researchers argue could be remnants of these primordial networks. Critics, of course, point out gaps. How did these sets transition from chemistry to genetics? Without replication, how could natural selection act? Yet Kauffman's ideas offer a compelling counterpoint. Maybe selection began at the network level, with entire sets competing for resources based on stability or efficiency. Some sets would persist, others collapse, a crude form of chemical evolution before genes took the reins. What's haunting about this theory is its universality. Autocatalytic sets don't require Earth-like conditions, just diversity, energy, and time. Titan's methane lakes, Europa's subsurface oceans, even the clouds of Venus could host similar networks, silently weaving complexity in the dark. It's a reminder that life's building blocks might not be molecules alone, but the invisible threads that connect them, the patterns that turn chaos into something capable of wonder. And in that shift, the universe reveals its quiet talent for finding order, even in the unlikeliest of places. Yellowstone's otherworldly landscapes, steaming pools of electric blue, geysers launching plumes of scalding water skyward, aren't just geological wonders. They might be time capsules preserving the very conditions where life first flickered into existence. The geothermal field theory suggests that terrestrial hot springs, far from being just scenic attractions, were primordial laboratories where Earth's earliest chemistry edged toward biology. Unlike deep-sea vents, these land-based systems offered a unique blend of features: silica-rich waters, dramatic temperature swings, and mineral surfaces that could have acted as both scaffolds and catalysts for life's first fragile assemblies. Picture a hot spring 4 billion years ago, its waters laden with dissolved silica, the same compound that forms quartz and opal. As the water cools along the spring's edges, the silica precipitates out, creating intricate microstructures with vast surface areas. These aren't just rocks, they're molecular sieves, they're nooks and crannies perfectly sized to trap and concentrate organic molecules. Experiments show that silica surfaces can accelerate the formation of peptide chains of amino acids by orders of magnitude. Even more strikingly, silica's charged surface can align molecules in specific orientations, almost like a chemical assembly line. It's as if the geology itself was nudging randomness toward order, offering a stage where lifeless chemistry could rehearse for biology. But silica wasn't just a passive stagehand. Hot springs create dynamic gradients, scalding at the center, lukewarm at the edges, that could have driven molecular evolution. Imagine a molecule forming near the spring's boiling heart, then being swept outward as the water flows. The cooling temperature might stabilize it, allowing it to persist long enough to interact with other compounds. Over countless cycles, this natural sorting mechanism could have selected for increasingly complex molecules, favoring those that could withstand the spring's ever-shifting conditions. Modern hot springs support this idea. They're teeming with extremophiles, microbes that thrive in conditions lethal to most life, hinting that these environments have long been crucibles for chemical innovation. Energy, too, was abundant. Hot springs are natural batteries, their mineral-rich waters humming with electrochemical potential. Sulfur compounds, iron, and hydrogen sulfide, all common in these settings, could have fueled primitive metabolic cycles. Some researchers have recreated these conditions in labs, mixing silica, sulfides, and simple organics under geothermal temperatures. The results, not just random reactions, but patterns resembling modern metabolic pathways, as if the springs were running a crude version of life's operating system long before cells existed. What sets geothermal fields apart is their duality. They're both chaotic and nurturing, destructive and creative. A molecule near a boiling vent might be torn apart by heat, only for its fragments to recombine in cooler zones, forging something new. This push and pull mirrors life's essence, a constant dance between order and disorder, persistence and change. And because terrestrial hot springs likely existed on other rocky worlds, like Mars or even ancient Venus, the theory suggests life's recipe might not be as Earth-exclusive as we once thought. The geothermal field theory doesn't just answer how life might have started, it whispers why. In these steamy, mineral-laden waters, the planet wasn't just mixing chemicals, it was testing resilience, rewarding adaptability, and sculpting complexity through relentless natural experiments. Every bubbling pool, every mineral crust, every shimmering steam plume becomes a testament to the universe's quiet determination to turn chemistry into something more. And if that's true, then Yellowstone's iconic geysers aren't just geological wonders, they're living monuments to the most improbable yet inevitable transformation in cosmic history. Phosphorus might not get the same attention as carbon or oxygen, but without this unsung hero of the periodic table, life as we know it simply wouldn't exist. Every time your cells burn energy, every time your DNA replicates, phosphorus is there, the silent workhorse tucked into the backbone of ATP, the universal energy currency, and RNA, life's original information molecule. Yet here's the paradox: while phosphorus is essential, it's also notoriously scarce in forms life can use. On early Earth, most phosphorus was locked away in insoluble minerals, barely accessible to primitive chemistry. So how did biology crack this puzzle? The answer might lie in celestial interventions and cosmic luck, reshaping how we see life's most stubborn bottleneck. One radical idea is phosphorus panspermia, the notion that Earth's phosphorus didn't just come from its crust but rained down from space. Meteorites like the Yamato asteroid fragments contain phosphide minerals which react violently with water to release phosphites, soluble compounds that could have fueled early biochemistry. Even more compelling, the Murchison meteorite carried phosphonates, rare organophosphorus molecules that resemble the phosphate esters in ATP. These discoveries suggest space wasn't just sprinkling Earth with raw ingredients. It might have delivered them pre-packaged in biologically useful forms. Imagine a young planet, its oceans still sterile, being seeded by carbon-rich asteroids bearing gifts of reactive phosphorus, a cosmic care package that tipped the scales toward life. But meteorites weren't the only possible delivery system. Volcanic activity could have unlocked Earth's native phosphorus through sheer geologic grit. When lightning strikes volcanic ash, it creates fulgurites, glass-like tubes where phosphorus compounds concentrate. Hydrothermal vents, too, might have played alchemist, transforming stubborn apatite into more soluble forms under high pressure and heat. Recent experiments show that iron-rich minerals common in vents can catalyze the conversion of phosphate into pyrophosphate, a primitive cousin of ATP's energy-carrying potential. It's as if the planet itself was experimenting with workarounds, jury-rigging solutions to its own phosphorus shortage. The stakes of this scarcity can't be overstated. Modern cells invest enormous energy to scavenge and recycle phosphorus, a hint that early life faced the same relentless pressure. Some scientists argue this constraint wasn't just a hurdle but a creative force. Scarcity may have driven life to adopt RNA as its first genetic material instead of DNA, simply because RNA uses half the phosphorus. It's a testament to biology's frugality. When the universe gives you lemons, you build a genome that squeezes every drop. Yet the phosphorus puzzle also raises a tantalizing question. If life here had to rely on meteoritic handouts or geologic flukes, does that make Earth's story unique? Not necessarily. Mars, for instance, shows evidence of ancient phosphorus-rich hot springs. Europa's icy shell might concentrate phosphorus in subsurface liquid pockets. The challenge of scarcity could be universal, but so might the solution's cosmic deliveries, volcanic ingenuity, or even exotic chemistries we've yet to imagine. What's striking is how phosphorus's role flips the script on life's origins. We often focus on the abundance of ingredients, but sometimes it's the limitations that shape everything. Life didn't just emerge because the conditions were perfect. It fought for every atom of phosphorus, turning a cosmic shortage into a catalyst for innovation. And in that struggle, we find one of biology's oldest lessons. Scarcity doesn't stifle life, it sharpens it. Life's molecules have a peculiar quirk. They're picky about their symmetry. Every amino acid in your body twists to the left, while every sugar in your DNA coils to the right. This universal preference, called homochirality, is one of biology's strangest puzzles. Why would nature favor one mirror image over the other when both forms should be equally likely? The answer might hold the key to understanding how lifeless chemistry first crossed into biology. Chirality comes from the Greek word for hand, and like your hands, molecules can be mirror images that refuse to perfectly overlap. Most chemical reactions produce equal amounts of left and right-handed versions, a 50-50 split. Yet life stubbornly chooses one. This bias isn't just a curiosity, it's essential. Enzymes, the molecular machines that power your cells, rely on precise shapes to function. A right-handed enzyme won't fit a left-handed amino acid, just as a left-handed glove won't fit your right hand. If early life had mixed chirality, its chemistry would have sputtered into chaos. So how did nature settle on left for amino acids and right for sugars? One theory points to the stars literally. In the 1990s, scientists analyzed the Murchison meteorite and found a slight excess of left-handed amino acids. This cosmic hint suggests that asymmetric molecules might have arrived pre-packaged from space. The culprit, polarized starlight. In interstellar clouds, ultraviolet light spirals as it passes through magnetic fields, and this twisted light can selectively break one chiral form over the other. Experiments show that irradiating amino acids with polarized UV light creates small but consistent imbalances. Over billions of years, these tiny biases could have snowballed, with meteorites delivering their skewed cargo to Earth's primordial soup. But space isn't the only possible architect. Earth itself might have tipped the scales through crystal power. Certain minerals, like quartz, form chiral surfaces at the atomic level. When amino acids latch onto these crystals, they can align in matching orientations, amplifying a slight initial imbalance into a dominant form. In a groundbreaking experiment, researchers coated chiral quartz crystals with amino acids, then washed them with water, mimicking tidal cycles. The result, one chiral form persisted while the other washed away. This crystal-induced symmetry breaking could have acted like a molecular filter, sieving randomness into order drop by drop. There's also a darker possibility, chance. Some models suggest that early Earth's chemistry might have started with near-perfect symmetry, only for a random fluctuation, a tiny excess of one form, to snowball through autocatalytic reactions. Once a small lead was established, self-reinforcing cycles could have locked in the bias like a ball rolling downhill. This idea, called the Frank model, shows how fragile the balance might have been. Flip the initial conditions and life could have ended up with the opposite chirality, right-handed proteins, left-handed sugars. The implications ripple far beyond Earth. If polarized starlight or chiral crystals set the rulebook, then life elsewhere might share our preferences. But if chance ruled, alien biochemistry could twist the opposite way. Future missions could look for chirality in Martian soil or Europa's plumes. A finding of left-handed amino acids there would hint at universal processes, while right-handed ones would suggest a cosmic coin toss. What's haunting is how this tiny asymmetry echoes through biology. Your body rejects wrong-handed molecules. Thalidomide's tragic side effects showed how one chiral form can heal while the other maims. Even our sense of smell distinguishes mirrored molecules. One version smells of oranges, the other of turpentine. Chirality isn't just a footnote in life's story, it's the signature on every page. The mystery endures because it's more than a chemical quirk. It's a window into the moment when randomness crystallized into inevitability, when the universe stopped flipping a coin and chose a direction. Whether by starlight, stone, or sheer luck, that choice shaped everything that followed, a reminder that life's foundations are as elegant as they are enigmatic. Black smokers, the deep-sea chimneys belching superheated, mineral-rich plumes into the ocean's darkness, seem like the last place life would take root. Unlike their gentler alkaline cousins, these towering vents spew water as hot as 700 degrees Fahrenheit. laced with sulfur and heavy metals, a broth so acidic it can dissolve solid rock. Yet within this inferno lies a paradox. These brutal conditions might have forged some of life's earliest chemical breakthroughs. The black smoker hypothesis centers on their unique architecture. As scalding, mineral-laden fluids gush from Earth's crust, they meet frigid seawater, causing iron and sulfides to precipitate into hollow, porous chimneys. These structures aren't just geological oddities, they're dynamic reactors. Their walls, riddled with microscopic compartments, create countless niches where molecules could concentrate and interact. Crucially, the chimney's iron sulfide minerals pyrite and mechanowite aren't just bystanders. They're chemists. Lab experiments show these minerals can catalyze reactions that turn carbon dioxide and hydrogen into acetic acid, a cornerstone of metabolism. Some even form tiny, cell-like bubbles, their mineral walls mimicking the lipid membranes of modern cells. Temperature, though, remains the elephant in the room. How could delicate organic molecules survive such heat? Here's the twist: black smokers aren't uniformly scorching. Their outer edges, where vent fluids mix with cold seawater, hover around a balmy 70 to 100 degrees, a Goldilocks zone where heat energizes reactions without obliterating them. This gradient matters. Imagine a molecule forming in a cooler crevice, then being swept inward by convection currents, gaining energy to react before cycling back to stabilize. It's nature's own PCR machine, thermal cycling driving chemistry forward. Acidity poses another hurdle. Black smoker fluids can be as corrosive as battery acid, a death sentence for most modern biomolecules. But life's pioneers might have turned this poison into a tool. Sulfide minerals can buffer pH, creating microenvironments where acidity fluctuates, a push-pull that could have selected for molecules tough enough to handle stress, a trait life never outgrew. Today's extremophiles, like the acid-loving archaea in Yellowstone's hot springs, prove biology can tame even the harshest chemistries. Yet the black smoker hypothesis isn't without its skeptics. Critics note that while iron sulfide chemistry is robust, it struggles to explain life's leap to genetic coding. RNA, for instance, degrades rapidly in high heat and low pH. Some researchers propose hybrid scenarios. Perhaps black smokers birthed metabolism, while cooler alkaline vents later hosted genetics with molecules shuttling between environments. Others point to fossilized vent microbes in ancient rocks, though debates rage over whether these are truly biological or mere mineral mimics. What's undeniable is how these vents reshape our imagination. Life didn't necessarily need gentle cradles. It might have emerged in a pressure cooker, forged in the same fires that once made Earth hellish. If black smokers did play a role, it suggests life's beginnings weren't fragile but ferocious, a testament to biology's ability to thrive where even the stoutest steel would rust. And that changes how we search for life beyond Earth. Jupiter's moon Io, with its volcanic sulfur lakes, or Saturn's Enceladus, where hydrothermal activity may churn beneath icy crusts, could be running similar experiments right now, proving once again that life, given the right chemistry, will find a way to burn bright even in the darkest depths. Long before ATP became biology's universal energy currency, another molecule might have powered life's earliest reactions: polyphosphate, a chain of linked phosphate groups that packs a formidable energetic punch. Picture volcanic landscapes on early Earth, where scorching plumes of gas condensed into mineral crusts rich in these phosphorus chains. Unlike the delicate dance of ATP synthesis, polyphosphates are rugged survivors, stable enough to withstand extreme heat, yet reactive enough to fuel the kind of crude biochemistry that predated sophisticated cells. This is the polyphosphate world hypothesis, a bold reimagining of life's first power source that challenges one of biology's most fundamental assumptions. The case for polyphosphates starts with their sheer simplicity. ATP, while efficient, requires an intricate machinery of enzymes to synthesize and recycle. Polyphosphates, by contrast, form spontaneously under volcanic conditions. When phosphorus-rich gases like phosphine erupt from magma and meet water vapor, they condense into polyphosphates, a process replicated in labs mimicking early Earth's geology. These chains act like molecular batteries, their high-energy phosphate bonds primed to transfer energy with minimal fuss. Modern organisms still use polyphosphates as backup generators. Bacteria stockpile them to survive starvation, and human bone cells rely on them to regulate mineralization. These lingering roles hint at an ancient inheritance, like evolutionary echoes of a time before ATP took over. But polyphosphates offer more than just energy, they're multitaskers. In test tubes, they catalyze the formation of peptides and nucleic acids, acting as both fuel and matchmaker for life's building blocks. Some researchers have even coaxed polyphosphates into driving primitive metabolic cycles, like the synthesis of acetyl phosphate, a molecule thought to predate ATP in energy metabolism. Their versatility extends to structure, too. Polyphosphates can bind metals like iron and zinc, creating complexes that resemble the active sites of modern enzymes. It's as if these molecules were nature's first Swiss Army knives, capable of juggling energy transfer, catalysis, and mineral assembly long before specialized proteins evolved. The volcanic connection seals the deal. Early Earth's surface was a patchwork of geothermal activity, with fumaroles and hot springs leaching polyphosphates into surrounding waters. These environments would have provided more than raw materials, they offered gradients. As polyphosphate-laden fluids mixed with cooler, organic-rich pools, the resulting energy disparities could have driven primitive versions of chemiosmosis, the same proton-powered process modern cells use to make ATP. In this view, life didn't invent energy harnessing from scratch. It co-opted a geochemical phenomenon that volcanoes had already been running for eons. Yet the theory isn't without its skeptics. If polyphosphates were so plentiful, why did life switch to ATP? The answer might lie in precision. ATP's single phosphate group allows for finer energy control, like trading a sledgehammer for a scalpel as biochemistry grew more complex. Others argue that without fossils of early phosphate chemistry, the hypothesis remains speculative. Still, the polyphosphate world reshapes how we see life's rise, not as a sudden spark, but as a slow handoff from geology to biology, where the planet itself supplied the first sparks of vitality. This idea also whispers tantalizing clues for astrobiology. Jupiter's moon Io, with its violent volcanic plumes, or Saturn's Enceladus, where geothermal vents churn beneath icy crusts, could be crafting polyphosphates right now. If life's first energy source was born from fire and rock, then the universe might be quietly brewing the same recipe wherever magma meets water, and that changes everything. The search for life's origins isn't just about finding the right molecules, it's about listening to the whispers of planets as they murmur the secrets of turning stone into biology. High above ancient Earth's turbulent surface, where storm clouds gathered and lightning split the sky, another crucible for life's building blocks may have been quietly at work, not in oceans or vents, but on the countless microscopic particles floating through the atmosphere. The gas-particle reaction theory proposes that dust, salt crystals, and aerosols acted as tiny laboratories, their surfaces providing the perfect stage for simple gases to collide, react, and assemble into complex organic molecules before raining down into the waiting oceans below. These atmospheric particles weren't just passive specks, they were alchemists. Their surfaces, often rich in metals or minerals, could have catalyzed reactions between gases like carbon monoxide, methane, and ammonia, chemicals abundant in Earth's early air. Lab experiments reveal the astonishing potential of this process. When scientists simulate primordial atmospheres in reaction chambers, introducing UV light or electrical discharges to mimic sunlight and lightning, something remarkable happens. In the presence of silicate or clay aerosols, simple gases transform into amino acids, sugars, and even nucleobases, the very molecules life uses to store information and build structures. Without the particles, these reactions proceed sluggishly or not at all. But with them, complexity emerges at a staggering pace, as if the dust itself were guiding chemistry toward biology. The magic lies in surface chemistry. Aerosols provide something the open air can't, a physical scaffold where molecules linger long enough to interact. On a flat surface, a carbon monoxide molecule might briefly meet a nitrogen molecule before drifting apart, but trap them in the microscopic crevices of a clay aerosol and their chances of reacting skyrocket. Some minerals, like titanium dioxide, even act as photocatalysts using sunlight's energy to turbocharge reactions, much like chlorophyll does in plants today. It's a delicate dance of chance and necessity, the right particles, the right gases, and the right energy all converging in the sky. What makes this theory so compelling is its universality. Earth's early atmosphere wasn't unique. Similar hazes shroud Saturn's moon Titan and likely enveloped Venus in its youth, even interstellar space teams with dust-coated organics. If aerosol chemistry could brew life's precursors here, why not elsewhere? The implications stretch beyond origins. Modern smog particles, eerily enough, host complex organic reactions, proving the atmosphere never stopped being a chemical reactor, it just changed cooks. Yet mysteries remain. How did these sky-borne molecules survive the descent to Earth's surface without being destroyed by UV radiation or heat? Some researchers point to protective mineral coatings or the shielding effect of water droplets in clouds. Others suggest that the molecules didn't need to survive intact. Even fragments could have reassembled in the oceans, like scattered puzzle pieces finding their way back together. The gas particle theory doesn't just add another location to life's origin story, it expands the stage entirely. No longer confined to deep sea vents or tidal pools, life's beginnings might have been written in the clouds, scribbled onto specks of dust, and signed with a drop of rain. And in that shift, we're reminded that the universe's creativity knows no bounds, turning even the air we breathe into a cradle for the impossible. Earth's magnetic field might seem like an invisible force we take for granted, but without its protective embrace, life as we know it could have been snuffed out before it even began. This unassuming shield, generated by the churning of molten iron in Earth's core, deflects the full fury of solar radiation particle storms that would otherwise strip away atmospheres and scald surfaces sterile. For fragile prebiotic molecules trying to assemble into something more complex, this magnetic barrier could have meant the difference between survival and obliteration. Consider the violence of our young solar system. 4 billion years ago, the sun was a tempestuous toddler, unleashing solar flares hundreds of times more powerful than today's. Ultraviolet radiation blasted Earth's surface unchecked, since the ozone layer didn't yet exist. Without protection, any promising chemical experiments unfolding in tidal pools or volcanic springs would have been shredded by this relentless assault. But Earth had a hidden advantage, a magnetic dynamo already humming in its core courtesy of its rapid rotation and molten interior. Evidence from ancient zircon crystals suggests this field was active as early as 4.2 billion years ago, right when the first whispers of prebiotic chemistry were likely underway. The field's importance becomes stark when we compare Earth to Mars. The red planet once had flowing water and possibly even organic chemistry, but its smaller size allowed its core to cool, shutting down its magnetic dynamo. Without that shield, solar winds gradually stripped away Mars' atmosphere, leaving its surface barren. Earth, by contrast, maintained both its atmosphere and its liquid water, a stable laboratory where delicate molecular experiments could run uninterrupted for millennia. Some researchers argue this stability was more than just helpful, it was essential. RNA molecules, for instance, are notoriously vulnerable to UV damage. Even with tidal cycling or mineral shielding, persistent radiation would have made accumulating complexity nearly impossible without a planetary-scale deflector. Timing is everything in this theory. Paleomagnetic studies reveal our magnetic field wasn't always equally robust. During the late heavy bombardment around 3.9 billion years ago, when asteroids pummeled Earth's surface, the field might have flickered only to strengthen afterward. Intriguingly, this coincides with estimates for when life first emerged. Did the calm after the cosmic storm, paired with a reinforced magnetic shield, create the perfect window for life's fragile chemistry to finally take root? The correlation is tantalizing if still circumstantial, yet the magnetic field wasn't just a passive protector. Some scientists propose it actively shaped life's trajectory. Charged particles guided along magnetic field lines could have concentrated at the poles, driving atmospheric chemistry that created complex molecules. Others suggest that early microbes might have evolved to sense magnetic fields, using them as navigational aids, a trait still seen in magnetotactic bacteria today. This hints at a deeper dialogue between Earth's geophysics and biology, where the planet didn't just host life but actively collaborated in its creation. The implications stretch far beyond our own origins. As we discover exoplanets, we're learning that magnetic fields aren't universal. Super-Earths might lack them if their high pressures solidify their cores, while tidally locked planets could have lopsided fields. A planet's magnetic heartbeat or its absence might be one of the most overlooked factors in its habitability. Our search for alien life often focuses on water and atmosphere, but perhaps we should also ask, does it hum with a magnetic song? After all, Earth's story suggests that life isn't just about having the right ingredients, it's about keeping them safe long enough to work their magic. Imagine a comet, a frozen time capsule from the solar system's birth, streaking through Earth's early atmosphere and splashing into a shallow coastal pond. The impact doesn't just bring water, it delivers a payload of complex organic molecules forged in interstellar space. Now picture the tide rolling in, diluting this cosmic broth with mineral-rich seawater, then receding under the sun's warmth to concentrate the mixture. This rhythmic dance of dilution and evaporation, combined with the comet's extraterrestrial ingredients, could have created the ultimate probiotic laboratory. Welcome to the cometary pond theory, where two of life's origin stories, panspermia and tidal cycling, collide in a single scenario more potent than either alone. Recent missions like Rosetta and Stardust have transformed our understanding of cometary chemistry. When the Philae lander analyzed Comet 67P's surface, it found a stunning array of organics, from glycine, the simplest amino acid, to phosphorus, a key element for DNA and energy molecules. Even more remarkably, these compounds weren't scattered randomly, they clustered in microscopic icy grains, their structures preserved by the vacuum of space for billions of years. Laboratory experiments simulating comet impacts show that when these icy grains hit shallow water, they release their molecular cargo intact, as if the universe had pre-packaged life's ingredients in freezer-safe containers. But here's where the tidal pool connection becomes crucial. A deep ocean would dilute these precious molecules into oblivion, while an isolated puddle might lack the minerals to catalyze reactions. Shallow coastal ponds, however, offer the perfect middle ground. Their fluctuating water levels act like a natural evaporator, concentrating cometary organics during dry phases, while wave action and mineral sediments provide catalytic surfaces. Studies at volcanic lagoons in Iceland, environments resembling early Earth, demonstrate this beautifully. When researchers added comet-like organics to these pools, the combination of wet-dry cycles and volcanic minerals produced peptide chains up to 20 amino acids long, orders of magnitude faster than static solutions. The theory also solves a persistent panspermia puzzle: delivery without destruction. While high-velocity impacts vaporize organic payloads, a comet grazing the atmosphere at low angles or fragmenting into smaller pieces could deposit its cargo gently. Some models suggest up to 30% of a comet's organic content might survive such an arrival, especially if cushioned by water, and coastal ponds, often lined with clay or silica gels, would provide further protection, their mineral surfaces acting like molecular Velcro to catch and preserve fragile compounds. What emerges is a best-case scenario where cosmic and terrestrial processes cooperate. The comet brings the building blocks, the pond provides the workshop. This synergy might explain why life appeared so rapidly once Earth cooled. Perhaps it wasn't starting from scratch, but assembling a puzzle where half the pieces arrived pre-cut. As we search for life beyond Earth, this hybrid model expands our horizons. Europa's icy crust, pockmarked with reddish deposits that might be organic rich comet strikes, or Mars' ancient shoreline basins could have hosted similar experiments. The cometary pond theory doesn't just reshape our past, it gives us a new template for where to hunt next in the cosmos. Ultimately, this idea celebrates life's resourcefulness. It suggests the universe doesn't need perfect conditions to spark biology, just a chance encounter between the right traveler and the right environment. A comet's frozen chemistry meeting Earth's dynamic shores becomes more than an impact. It's a handshake across the stars, the moment when two cosmic processes conspire to turn chemistry into something far more extraordinary. Before RNA became the star of life's origin story, another molecule might have stolen the show: peptide nucleic acid, or PNA. Unlike RNA's delicate sugar phosphate backbone, PNA is built from peptides, the same sturdy chains that form proteins, making it far more rugged in the harsh conditions of early Earth. Picture a molecule that combines the information storage prowess of DNA with the durability of a protein, capable of forming stable double helixes without needing the precise conditions RNA demands. This hybrid could have been the universe's first genetic material, bridging the gap between simple chemistry and the complex coding of life. Lab experiments reveal just how plausible this idea is. Under conditions mimicking Earth's primordial environments, think volcanic vents or tidal pools, scientists have synthesized PNA strands from simple starting materials like amino acids and nucleotide bases. One groundbreaking study showed that PNA can self-assemble at temperatures ranging from freezing to near boiling, a flexibility RNA can't match. Even more astonishing, PNA strands can spontaneously pair up, forming helical structures that mirror DNA without requiring enzymes or external energy. It's as if nature had a backup plan, a molecule that could store information without the fussiness of RNA's sugar chemistry. The stability of PNA isn't just a lab curiosity, it's a game changer for origin theories. RNA's Achilles heel is its fragility. It breaks down rapidly in acidic or high-temperature environments, exactly the conditions found near hydrothermal vents or volcanic springs. PNA, by contrast, laughs off these challenges. Its peptide backbone resists hydrolysis, meaning it could have persisted long enough to replicate and evolve in places where RNA would have disintegrated. Some researchers argue this durability makes PNA the perfect missing link, a molecular workhorse that laid the groundwork for RNA's later takeover. But PNA's talents don't stop at survival. It's also a gifted mimic. Modern cells rely on RNA's ability to pair with DNA for transcription, and PNA can do the same, binding to both DNA and RNA with even greater affinity than natural nucleic acids. This suggests PNA could have acted as a universal adapter, facilitating the transition from a protein-dominated world to one governed by genetic coding. In test tubes, PNA strands have been coaxed into directing the assembly of RNA molecules, hinting at a handoff between these two systems that might have been critical for life's ascent. Yet the PNA world hypothesis isn't without its skeptics. If PNA was so versatile, why did life switch to RNA at all? The answer might lie in RNA's catalytic abilities. While DNA excels at stability and information storage, it lacks the dynamic folding that lets RNA act as both template and catalyst. Some scientists propose a hybrid scenario. DNA provided the initial genetic framework, while RNA's multitasking prowess eventually won out. Others point to the lack of DNA in modern organisms, though traces of its chemistry might linger in the way certain proteins interact with nucleic acids today. What's electrifying about this theory is how it reshapes our search for life's origins and its future. Synthetic biologists are already exploring DNA as a tool for genetic engineering, capitalizing on its robustness to create new forms of artificial life. Meanwhile, missions to icy moons like Europa might look for DNA-like molecules in subsurface oceans where stability trumps complexity. The RNA world doesn't just rewrite life's past, it opens doors to possibilities we're only beginning to imagine, a testament to the universe's relentless ingenuity in turning simplicity into sentience. Titan, Saturn's largest moon, is a world straight out of science fiction, its thick orange haze hiding vast seas not of water, but of liquid methane and ethane, where temperatures plunge to -290 degrees Fahrenheit. In this frigid alien landscape, the rules of chemistry rewrite themselves, opening the door to a radical possibility. Life might not need water at all. The hydrocarbon seas theory dares us to imagine biology built from entirely different blueprints, where methane plays the role of water, and the molecules we consider essential for life are swapped for frost-resistant alternatives. On Earth, water's unique properties make it the perfect solvent for life. It dissolves nutrients, facilitates chemical reactions, and provides a stable medium for cellular structures. But on Titan, methane takes center stage. Though it lacks water's polarity, methane's low surface tension allows it to seep into porous materials, potentially creating microscopic environments where molecules could interact. Ethane, another abundant liquid on Titan, could act as a solvent for nonpolar compounds, setting the stage for chemistry that would be impossible in Earth's watery oceans. The Cassini-Huygens mission found evidence of complex organic molecules in Titan's atmosphere, including acrylonitrile, a compound capable of forming stable membranes in methane, akin to the lipid bilayers of our own cells. The real mind-bender isn't just the solvents, it's the potential biochemistry. Instead of DNA, life on Titan might rely on polyamines, chains of nitrogen and carbon that remain flexible even at cryogenic temperatures. These molecules could store genetic information while withstanding Titan's deep freeze. Energy might come not from sunlight, which barely penetrates the moon's haze, but from acetylene or hydrogen reacting with atmospheric nitriles, a metabolic pathway as alien to us as photosynthesis would be to Titan's hypothetical organisms. Some scientists propose that the moon's cryovolcanoes, spewing water ammonia slurries, could create transient warm pockets where reactions akin to Earth's hydrothermal vent chemistry might occur, blending the familiar with the exotic. This theory shatters Earth-centric assumptions about life's requirements. If life can emerge in Titan's methane lakes, then the universe's habitable zones expand dramatically, no longer limited to the Goldilocks distance from a star, but encompassing any world with liquid reservoirs and energy gradients. It forces us to reconsider the very definition of life. Must it be carbon-based, water-dependent, or could it manifest in forms we've yet to recognize, thriving in conditions we'd consider utterly hostile? The implications ripple outward. Titan isn't alone. Pluto's subsurface oceans, Neptune's moon Triton, and even exoplanets with hydrocarbon atmospheres could host similar chemistries. Future missions like NASA's Dragonfly drone aim to probe Titan's surface for signs of probiotic molecules or even metabolic activity. Discovering evidence of a second genesis there would be among the most profound scientific revelations in history, proving that life isn't a fluke of Earth's particular conditions, but a cosmic inevitability, adaptable, resilient, and endlessly inventive. What's haunting about this idea is how it redefines our place in the universe. We've long wondered if we're alone, but the hydrocarbon seas theory suggests a stranger possibility. We might not even recognize life if it were right in front of us, whispering in a chemical language we're only beginning to decipher. Titan's methane waves rolling under a dim orange sky could be the first chapter in a story far richer and more bizarre than we ever imagined. What if life didn't begin in our universe at all? This is the spine-tingling premise of the multiverse life hypothesis, the idea that biology's first whispers might have echoed from an entirely different cosmos, one where the rules of physics themselves are rewritten. Imagine a reality where gravity is stronger, light moves slower, or the very atoms that make up matter behave differently. In such a place, life could have emerged under conditions impossible here, only to later hitch a ride through some crack in the fabric of space-time, seeding our universe with its strange, foreign blueprint. The multiverse theory itself isn't pure speculation, it's a legitimate framework in theoretical physics. Some models of inflation, like eternal inflation, suggest our universe is just one bubble in an endless froth of universes, each with its own unique physical constants. Most would be sterile, their laws too chaotic or rigid for complexity, but a rare few might strike a delicate balance, permitting chemistry and life to unfold in ways we can scarcely imagine. The real leap is the notion that these universes aren't entirely isolated. Hypothetical structures like Einstein-Rosen bridges, wormholes, or quantum foam fluctuations could, in theory, allow tiny transfers of matter or information between worlds. If even a single microbe or self-replicating molecule slipped through, it might have ignited life here without needing Earth to start from scratch. Philosophers have long been fascinated by the anthropic principle, the observation that our universe's constants seem eerily fine-tuned for life. But what if the answer isn't tuning at all? If countless universes exist, some must randomly hit the jackpot, and if life can spread between them, it transforms the puzzle. Suddenly, we're not special, we're just the beneficiaries of cosmic cross-contamination. The idea echoes Panspermia, but on a scale so vast it redefines the word alien. Yet for all its grandeur, the theory stumbles on one glaring problem: evidence. Wormholes, if they exist, are likely unstable or microscopic. Quantum tunneling between universes remains firmly in the realm of equations. And even if life could survive such a journey, detecting its extra-dimensional origins would require breakthroughs beyond today's science. Still, absence of proof isn't proof of absence. Some researchers point to statistical quirks like the unexpected scarcity of certain isotopes in Earth's crust as potential, if highly speculative, hints of foreign physics. Others argue that if life's building blocks are ubiquitous across the multiverse, their arrival here might leave no distinguishable trace. What's irresistible about this idea isn't its plausibility, but its audacity. It turns the search for life's origins into a cosmic detective story spanning not just planets or galaxies, but entire realities. If true, it means our DNA could carry fragments of a universe where stars burn differently, time flows backward, or matter itself has alien properties. It's a reminder that biology's roots might be deeper and stranger than we ever dared to dream, and that's the heart of science's greatest theories, not just answering questions but revealing how much we've yet to ask. Whether multiverse life remains a thought experiment or one day leaps from philosophy to fact, it challenges us to rethink the very boundaries of existence. After all, if life can cross the ultimate frontier, the walls between universes, then perhaps the only limit to its persistence is the breadth of our imagination. From scalding hydrothermal vents to icy cometary ponds, from the quantum dance of subatomic particles to the vast possibility of multiverses, we've journeyed through 30 breathtaking theories about how life might have first stirred in the cosmos. Each idea, no matter how wild or grounded, adds a crucial piece to the puzzle. The iron-sulfur world teaches us how metabolism could have emerged from bare rock. The RNA world shows how information might have gained a voice. Panspermia stretches our imagination across the stars, while cosmic dust reminds us that the universe itself could be the ultimate chemist. What's astonishing isn't just the diversity of these theories, it's how they intertwine. Maybe life didn't arise from one perfect scenario, but from a chorus of unlikely events. A comet delivers organic molecules, a tidal pool concentrates them, a hydrothermal vent provides energy, and RNA takes the reins. Or perhaps the truth lies in a theory we haven't even dreamed up yet. After all, every few years, a new discovery, a microbe surviving impossible conditions, a molecule performing an unexpected trick, reshapes what we thought was possible. This search for life's origins isn't just about the past, it's about our future. Every time we probe Europa's icy shell or simulate probiotic chemistry in a lab, we're not just solving an ancient riddle, we're learning how to recognize life wherever it might exist. And that's the real magic here. Whether life began in clay, in a volcanic spring, or on another world entirely, each theory expands our sense of what it means to be alive in this vast, mysterious universe. So what's your favorite idea? The elegance of the RNA world, the cosmic drama of panspermia, or something even more unexpected? Drop your thoughts in the comments. I'd love to hear which theory sparks your imagination. And if you're hungry for more mysteries, from the birth of black holes to the fate of the cosmos, hit that subscribe button, because the universe isn't done surprising us yet, and neither are we. Thanks for joining me on this wild ride through the greatest question of all. How did it all begin? Until next time, keep looking up and wondering.